My name is Barituziga Banuna, and I'm a member of the Daniel Group. Today, I will be presenting on my poster entitled Novel Membrane Mimetic Bioelectronic Device for Study of Plant Transmembrane Protein Transport. To give a brief background into my project, over the last couple of years, a growing interest has been placed on understanding the response of plants to climate change and environmental stressors. Due to their sessile nature, plants have been forced to develop a robust signal pathway to adjust to changes in their environment. A key actor in this is the plasma membrane. Plant membrane transport proteins are up and down regulated with changes in the environment and relay hormones, ligands, and essential nutrients. The study of membrane-bound transporters is integral towards understanding the cellular processes in plants, but technologies to do so on this front have continually lagged behind the mammalian counterparts. One example of one of these systems is the supported lipid bilayer, or SLB, which is a planar bilayer composed of native membrane materials that retains many of the important characteristics of the plasma membrane, being its fluidity, composition, and orientation. In combination with organic electronic transistors, electrochemical transistors, these have been used to electrically and optically investigate the transport of ions across mammalian transport proteins. Our aim is to demonstrate that this membrane on chip platform can be adapted to plant systems by studying the transport across a copper-specific transport protein in plants known as COP1 that maintains copper homeostasis. To do so, we isolated protoplasm from Arabidopsis thanalia via enzymatic digestion of the cell wall and overexpressed our GFP tag protein of interest. Protoplasms were exposed to a blooding solution, inducing the budding off of small membrane vesicles, and these vesicles were then reconstituted as a supported lipid bilayer by vesicle fusion with POPC, POPG, fusogenic liposomes. The formation of the supported lipid bilayer was optically characterized via fluorescence recovery after photobleaching, where R18 labeled SLBs were photobleached, and the intensity recovery data was used to determine the membrane diffusion coefficient. The supported lipid bilayers were also characterized electrically with electrical impedance spectroscopy, where the supported lipid bilayer is formed on top of a OACT and an alternating current is passed through at a range of frequencies, and the impedance response is then captured. The presence of a viable supported lipid bilayer can be determined by a characteristic behavior on a Bode plot and results in an increase in impedance as compared to a blank chip. Furthermore, the data from the EIS can be modeled as an equivalent electrical circuit as shown, in which the imparallel resistance, which is attributed to the SLB, is of interest. Successful transient and transfection of protoplasm was determined optically by fluorescence overlay, and COP1 GFP correctly localized to the plasma membrane, while the free GFP control was cytosolic. FRAP characterization showed that supported lipid bilayers created from both COP1 GFP and GFP control blebs were both mobile and could be fit to a 2D diffusion equation, indicating their planarity. Formation of the supported lipid bilayer was confirmed by EIS, where Bode plots of impedance versus frequency showed the characteristic chair behavior expected of a supported lipid bilayer, and disruption of the supported lipid bilayer with ethanol returned the values to at the baseline. This is seen in Figure A. Figure B shows that the concentration-dependent response of the supported lipid bilayer to a buffer containing copper ions, and shows that activation of the COP1 protein in the presence of copper results in a decrease in impedance. In this next figure, the normalized resistance values of the supported lipid bilayer is compared between COP1 GFP and GFP control at various copper concentrations. Both showed a concentration-dependent decrease in resistance with increase in copper concentration, but the response of the COP1 GFP was significant compared to the control. The presence of a response in the control could be attributed to endogenous expressed COP1 proteins, and an increased response in the overexpressed COP1 GFP samples is expected due to the higher concentration of the COP1s. In conclusion, we demonstrated that the membrane on chip platform can be successfully adapted to plant systems and can be used to study transport across transmembrane proteins in plants. This system can serve as a gateway to many new discoveries, and in the future may allow for similar studies of other membranes in the cell.